This recording is predominantly going to discuss meiosis, but just briefly, and it's going to talk about the difference between meiosis and mitosis. And the reason why I got to discuss meiosis a little bit is because in the process of talking about gametogenesis, which is the formation of gametes in the male and the female, you have to understand meiosis because the formation of the gametes requires meiotic divisions. In the female, the gametogenesis is referred to as oogenesis, and in the male, it is referred to as spermatogenesis. Now, the process of gametogenesis is a result of meiosis. Now, meiosis is a biological process by which we call diploid cells undergo cell division to form haploid cells. So I want to discuss diploid versus haploid first, and then we'll look at mitosis versus meiosis. Now, diploid and haploid refer to the number of chromosome sets a cell contain. If it's diploid, it has two complete sets of chromosomes. If it's haploid, it has half, like think HA, half the amount. It has one set. When we look at mitosis, the parent cell is diploid as well as the daughter cells. But in meiosis, the parent cell is diploid, but the daughter cells are haploid. So we're just going to look at mitosis first, just briefly. So in mitosis, for this example, just to make it for simplicity reasons, this cell has three sets of chromosomes. So you see set one, that's the black and the gray. Here's the set two, and here's the set three. Okay, so it has two complete sets. So we've got this one here. So we got a black and you got a gray, you got a red and you got an orange, you got a blue, you got a green. So each of these here are referred to as homologous chromosomes. So say, for example, the black and the gray, they are, we'll say chromosome 1. They're similar. It's where the homologous comes in. One was received from the dad or inherited from the dad. One was received from the mom. Okay, so on the chromosomes, they're going to have the same genes. Where there's going to be a difference is you may have some different variations or different alleles. Say, for example, we'll say these chromosomes contain a gene for eye color, and the one you got from the dad was a blue-eyed allele, and one you got from mom was a brown-eyed allele. The kid's going to be brown-eyed, just let give it to you. Brown-eyed is dominant over blue-eyed, but the chromosomes themselves will have the same genes along them. So we have here in the parent cell, we have a total of 6 chromosomes. You got these three sets these 3 pairs of homologous chromosomes. And what happens is the chromosomes will duplicate, and it will undergo one division. And after it undergoes one division, you'll have 2 cells, daughter cells, that have the identical number of chromosomes. Both cells are diploid. They're genetically identical. But in meiosis, it's a different story. You'd start off with a diploid cell, but after undergoing 2 meiotic divisions, you'll end up with 4 haploid gametes, which are not genetically identical. So we're going to go through this. So in meiosis, what we'll see is, again, we're starting off with these homologous chromosomes, which you see here. You will have DNA replication that occurs before meiosis 1. And what you'll see here is a tetrad formation. So say the black ones duplicate, so you see 2 of them here, you see the gray ones are 2 here, and you see it all the way across, but they kind of synapse here at the center, and we form a tetrad, which means 4. What happens is the synapsis of homologous chromosomes does occur during meiosis, and what we'll get is something called crossing over, where we'll have, say, I'm going to draw a picture here, is say we have, say, this black chromosome, got this gray chromosome next to it, is what can happen is we can have kind of some swapping of the DNA So you get genetic recombination. So you can get this genetic recombination because of crossing over. So this is what introduces the genetic variation. It's very important. So, but this picture actually doesn't show it to you, but that does occur. It does not occur during mitosis. What will happen here, which you'll see here, you've got that the tetrad formation. These cells are still diploid, and the cells are diploid because they still have two complete sets. So you still got a black, you got a gray, you got a red, you got a orange, you got a dark blue, you got a light blue. Now, after the first meiotic division, what you'll notice here, see this here, you see this here. This cell here does not have two complete sets. It's got black, it's got red, it's got dark blue. It doesn't have gray, orange, or blue, so it is haploid. So it does not have two complete sets. After the second meiotic division, you'll see here, where you have these kind of released, you don't have this anymore, is you have a total of 4 gametes that result. Each are haploid. So we started off with a cell that had a total of 6 chromosomes. The daughter cells, or these gametes, only have 3. So you see 3 in each of them, which can occur because let's do some math real quick. So you started off with 6 chromosomes. You had duplication of the DNA. That is 12. You did 2 meiotic divisions. So you got 6 divided by 2 again. You get 3. So each of the cells have 3 chromosomes. So each are haploid. Now the gametes here are going to be not genetically identical to the parent. And you don't know which of these different chromosomes are going to appear in each gamete. And remember, you can have a little different little of, so I'm just going to put some little gray in this one. And I'm going to put a little orange in this one here. So you have that genetic variation there with meiosis. So let's just briefly compare mitosis with meiosis again. So you both have DNA replication. So in mitosis, this takes place during interphase, something called interphase. In meiosis, it occurs before meiosis 1. Mitosis, you only have 1 division. In meiosis 2, you have 2. That's going to allow us to get to that haploid number. Mitosis, you do not, I'm going to say not applicable. There's no synapses of homologous chromosomes, but you do have it in meiosis. So yes, and you get crossing over. That is what introduces genetic variation. That's why my daughter is not a complete mix, or just her dad, just me, and she's a little bit different. So she's not, your children are identical to one or the other, or they can be a little bit different from both mom and dad. Now in mitosis, the number of daughter cells, you have 2, and they are diploid. And they are identical to the parent. In meiosis, you have 4 cells, which are haploid. We're going to call them gametes, and they are not identical to the parent. So what's the role of mitosis in the human body? Well, mitosis you need for normal cell growth and repair. So just say, for example, your epithelial tissues are always dividing. Say your skin cells, they're sloughing off. You're always producing more. So it's normal in the human body for cell growth and repair. But in meiosis, this is needed for gametogenesis, which is the formation of the gametes. And this is important, is it introduces genetic variation. So when you have a haploid spermatogonium, I keep saying spermatozoa, and a haploid oocyte, they meet up and become fertilized, or the egg, or the sperm fertilizes the egg, you're going to end up getting a diploid zygote when the DNA actually mixes together. So that's what we want. So this is the end of just mitosis versus meiosis. Look for the recordings about spermatogenesis and oogenesis / ovarian cycle, and also you can find recordings about the uterine cycle.